So, from here on we will discuss the major components of the tissue engineering triad, that is the materials, the cells and the signals required for skin tissue engineering. So, biomaterials that are used for skin tissue engineering can be classified as natural, synthetic and a combination of the two. So, we will discuss each one of them in detail. Natural polymers that are used for skin tissue engineering can be polysaccharide based polymers or protein based polymers. These are some examples of natural polymers that are used for skin tissue engineering. This is however not an exhaustive list. So, alginates, chitosan, cellulose, hyaluronic acid, dextran, pullulan, carrageenan, konjac glucomannan and isabgol are newer polymers that are being explored for skin tissue engineering. Protein based polymers such as collagen, gelatin, silk fibroin, keratin and elastin are being explored for skin tissue engineering. We will look at each one of them in detail. So, in natural polymers, chitosan is a very popular material used for skin tissue engineering, obtained from chitin, which is found in the cell wall, fungal cell wall and also the exoskeleton of insects. So, chitosan can be processed into different types of matrices such as films, nanofibers and sponges. So, several studies have been done using chitosan. So, here it shows an in vivo study where chitosan has been shown, it is highly biocompatible and has been shown to accelerate wound healing. And also chitosan has helped improve cellular adhesion and proliferation. Collagen is a protein based polymer. It is the major portion of the extracellular matrix, the ECM. Collagen also can be processed as nanofibers or 3D printed constructs. So, here you can see that using a collagen skin substitute, an acceleration in wound healing has been achieved in both freeze dried skin substitutes and electrospun skin substitutes. Reduced wound contraction and also a presence of hallmark of morphogenesis and cohesion, which shows the formation of the natural skin tissue has been observed. Silk fibroin is another natural polymer. Here it has been processed into nanofibers using electrospinning. Silk fibroin, electrospun silk fibroin has shown improved pore size and interconnectivity. The improved pore size and interconnectivity helps in good cellular infiltration and proliferation. Like standalone natural polymers, hybrid of two or more natural polymers have also been used, such as silk keratin scaffolds. So, here the keratin component which contains RGD sequences helps in improved cellular adhesion and proliferation than compared to having silk fibroin scaffolds alone. The blended material showed superior properties. Also, collagen, pullulan hydrogels have been synthesized. Collagen is usually used to make hard biomaterials. To make softer biomaterials, it is often blended with other natural polymers to make hydrogel systems. So, pullulan is one such example. So, in this hydrogel they obtained an open porous structure with improved cell viability and also achieved improved wound closure and vascularization. So, methacrylated gelatin and methacrylated hyaluronic acid have been blended, both natural polymers. Hyaluronic acid is also an ECM component. Gelatin is a natural protein based polymer, but both the polymers have their own disadvantages, such as hyaluronic acid does not help with cell adhesion and gelatin is poor in its mechanical properties. So, the blend of the two can help us overcome these shortcomings, and also physical or chemical modification of naturally occurring polymers can help improve their properties. So, here in this example the blend has been processed into a hydrogel, and they have embedded adipose derived stem cells into the material for improved vascularization. So, konjac glucomannan keratin. So, here konjac glucomannan is a polysaccharized based polymer, and keratin is a protein based polymer. So, the two have been blended to form scaffolds. These scaffolds they have done in vivo study where it showed enhanced collagen synthesis and deposition in diabetic wounds, and also the scaffolds have been shown to be highly biocompatible. Also other natural polymers have been blended together to have superior properties as skin tissue engineered products such as isabgol silk fibroin, collagen fibrin bioprinted constructs, or collagen chitosan scaffolds. So, now we look at synthetic polymers which have also been used for skin tissue engineering. Polyurethane, PVA, that is polyvinyl alcohol, PEG, polyethylene glycol, polyhema, polyhydroxyethyl methacrylate, and PLGA, polylactic glycolic acid, and polycaprolactone. Polyurethane is a very popular synthetic polymer used for skin tissue engineering. Several commercially available wound dressings are there which are made from polyurethane. So, this is an example of a polyurethane foam. Also you can load different bioactive agents into the materials to render better wound healing properties. So, here they have blended, they have added silver and asiaticoside into the polyurethane material and tested wound closure in a porcine model. So, here the material has shown to be biocompatible as seen as there is no skin irritation, and also they have observed faster wound healing in deep dermal wounds. So, PLGA is a copolymer of lactic acid and glycolic acid. So, here in this example they have synthesized nanoparticles, and they have loaded curcumin, a bioactive agent. So, they have observed reduced inflammation, increased reepithelialization, and increased collagen deposition, which are very important for wound healing. Polycaprolactone is another polymer. Here they have synthesized them as nanofibers using electrospinning. So, they have been embedded with placental derived bioactive molecules for wound healing. And polyhema is another synthetic polymer. They have synthesized it as a hydrogel loaded with moxifloxacin, and they have seen antioxidant activity and absence of inflammation. Also, like natural polymers, two or more synthetic polymers have been blended to achieve better properties. So, such as PCl and PEG block copolymer that has been synthesized and electrospun. So, here you can see the free amino groups to which they have attached epidermal growth factor, and they have done in vivo studies to show improved wound healing with epidermal growth factor loading. Also, there have been other synthetic polymer hybrids, PCl and PVA, curcumin loaded PCl and PVA nanofibers that showed improved wound healing. PEG and PLGA hydrogel, this was synthesized as a thermosensitive hydrogel or environmentally responsive hydrogel. This showed enhanced engraftment of muscle derived stem cells that were seeded onto the grafts. So, the combination of natural and synthetic polymers gives the gives better properties for skin tissue engineering. Natural polymers can provide biocompatibility to the graft, while synthetic polymers can provide the required mechanical strength. And also the natural polymers help with cell attachment and adhesion and proliferation. So, PVA CMC, that is a blend of polyvinyl alcohol and carboxymethyl cellulose, which is a carboxymethyl derivative of cellulose. Here PVA is the synthetic component and CMC is the natural polymer. So, PVA provides the mechanical properties to the material. PVA is however highly hydrophilic and it does not help with cell adhesion. So, carboxymethyl cellulose can help with cellular attachment and proliferation. So, the blend has been synthesized as a hydrogel, and in this particular study they have loaded reduced graphene oxide into the material and have found enhanced angiogenesis, which is crucial for wound healing. Silk fibroin PVA blend has also been used. Silk fibroin here is the natural component and PVA being the synthetic component. So, this has been fabricated as nanofibers. So, here you can see the silk fibroin based mat. It helps with cell recruitment, and also they have done in vivo studies to show improved wound healing and remodeling of the extracellular matrix. These are other combination of synthetic and natural polymers, PCL and collagen nanofibers. So, here they have loaded insulin delivering chitosan nanoparticles, which showed excellent in vivo wound healing, and also more than two components can be blended together to form skin tissue engineering materials. PVA starch chitosan has been used here to form a hydrogel and incorporated with nano zinc oxide to show excellent antibacterial activity and wound healing. So, skin substitutes can be classified as acellular and cellular matrices. The cellular matrices have cells loaded onto the graft. So, there are certain advantages to that. The cultured cells can deliver growth factors and ECM to the wounds, and also it has been shown that the cellular skin substitutes can show enhanced angiogenesis. So, what is the source of the cells that are used for seeding? So, they can be autologous, allogenic or xenogenic in as discussed previously, or they can be differentiated or stem cells. So, different cell types are used in skin grafts. They can be skin derived seed cells such as keratinocytes, which majorly occupy the epidermal layer, the dermal fibroblasts which lie in the dermis, the epidermal stem cells, and the melanocytes which provide pigment to the skin. Non-cutaneous cells such as embryonic stem cells, iPSCs, that is induced pluripotent stem cells, mesenchymal stem cells, and cells derived from the amnion have also been used. So, this is an image of keratinocytes cultured on electrospun chitosan nanofibers. Also, the seeding of cells can be done as a co-culture of two different type of cells. Fibroblasts and keratinocytes, for example, have been co-cultured, and it has been observed that by co-culturing them together, the fibroblast can actually help the proliferation of keratinocytes. Also, melanocytes have been co-cultured with fibroblasts and keratinocytes. This can help recreate the natural pigmentation process. Langerhans cells have also been co-cultured with fibroblasts and keratinocytes. So, this can help monitor skin immunological reactions, and dermal fibroblasts have also been co-cultured with endothelial cells. So, this can promote vascularization. Now, we are going to look at the third component of the tissue engineering triad, which are the signals. So, these signals can be biochemical or biophysical in nature. These signals provide functional and instructive matrix to allow skin regeneration. These signals can regulate cell matrix interactions, cell behavior and function. So, we can develop instructive materials rather than having passive grafts. These instructive materials can harness the body's innate ability to self repair. So, these are the different signals that can be given to the graft. So, some of these are biochemical signals that are growth factors and derivatives, the ECM proteins, small molecules, genetic regulators, and on the right you can see the biophysical signals such as the topography of the matrix, application of negative pressure, electric or electromagnetic stimulation. So, first we will look at biochemical signals. Biochemical signals can be growth factors. Growth factors are potent regulators of cellular activities such as cellular migration and proliferation and differentiation. Some growth factors that have been looked at for application in wound healing are vascular endothelial growth factor and epidermal growth factor and basic fibroblast growth factor and transforming growth factor beta. So, these play a potent role in all three phases of wound healing. So, conventionally these growth factors were developed were delivered using conventional approaches. These conventional delivery methods however cause burst release of the molecules. So, we are developing new techniques to deliver these molecules which can release the factors in a sustained manner and also so that they can be effective at a lower dosage. So, one such example can be attaching the growth factors to ECM proteins. Other than growth factors, there are derivatives and certain peptide sequences. The best characterized peptide sequence is the RGD sequence consisting of the amino acids arginine, glycine and aspartic acid. So, this sequence is found in several ECM proteins, and also these can be incorporated into synthetic materials which usually lack cell adhesion sites. This can help in cellular adhesion and migration into the graft. So, other molecules such small bioactive molecules can also be incorporated into the graft. So, one such molecule is oxygen. Oxygen levels have been found to be low in chronic wound tissues. Oxygen level is considered important because it provides energy for bacterial defense, for cell proliferation and also for cell migration. It is crucial. Oxygen has been used in hyperbaric oxygen therapy where it is delivered through systemic circulation. However, it fails to reach the desired site. So, then they started using oxygen as a topical therapy. Now, they have come up with oxygen delivering wound dressings such as Oxyzyme which releases oxygen through a chemical reaction. Another bioactive molecule that can be incorporated into tissue engineered grafts is nitric oxide. Nitric oxide is synthesized from nitric oxide synthase. This enzyme is present in 3 isoforms and all 3 isoforms have been found in the skin. In skin cells such as dermal fibroblasts, keratinocytes. So, this enzyme is present in 3 isoforms and 2 of the isoforms are constitutively expressed and another one is expressed by inducible expression. It is involved in various stages of wound healing, especially angiogenesis. This enzyme is very important for VEGF activity. Various genetic regulators have also been incorporated into tissue engineered grafts such as cDNA, small interfering RNA and microRNA. cDNA can be delivered using various non-viral vectors such as cationic polymers or liposomes or naked plasmids can be used. cDNA encoding various peptides and growth factors have been incorporated. One such protein is the sonic hedgehog protein. A study showed that the sonic hedgehog gene vector has been shown to improve diabetic wound healing with microvascular remodeling. Several small interfering RNA have been used. So, a small interfering RNA can cause silencing of disease causing genes by binding to the complementary sequences of the target mRNA. So, several diseases such as skin fibrosis have been treated using small interfering RNA. Another set of gene regulators are microRNA. These are endogenously found repressors that bind to the 3' untranslated regions of the mRNA. These microRNA have also been used to regulate angiogenesis and also involved in reepithelialization and tissue remodeling. Now we will look at biophysical signals. One such is the topography of the matrix. The topography of the matrix can guide cell migration through the graft. So, this can be achieved by using micro patterned surfaces. So, here you can see there is a PDMS membrane which has microfabricated grooves on it. So, which has been synthesized using a mold, and human dermal fibroblasts have been cultured on the membrane, and mechanical wounding is done on the surface, and you place your patch on the membrane and observe the cellular ingrowth. So, here you can see a video. So, in this video you can observe the difference in cellular dynamics between grafts that have perpendicular gratings and other grafts, blank grafts, that do not. So, here at 12 hours there is considerable ingrowth of cells into the graft where the perpendicular gratings are present, while in the blank graft even after 24 hours of wounding, the graft shows lower confluence compared to the one which has the gratings. Another biophysical signal that has been used is the application of negative pressure. So, this helps in increase in blood flow to the area and also helps in promotion of angiogenesis and granulation tissue formation. The mechanism that has been proposed here is when you apply the negative pressure, there is a slight deformation of the ECM which generates micro strain on the cells. This helps in cellular migration and proliferation. So, several vacuum assisted closure devices have been commercially made available. So, these devices mainly vary in their filler material that is used. So, either foam or antimicrobial gauze dressings have been used, and they also vary in the suction catheters that have been used. They also vary in the intensity of the negative pressure that has been applied, so which varies from 50 to 125 millimeters of mercury. So, in this figure you can see that on application of negative pressure there is exudant removal, edema reduction and blood supply increase that has been shown. Another biophysical signal used is electric stimulation. So, this is a biomimicry tool that has been used. It mimics the endogenous electric fields that arise after wounding and cause keratinocyte migration. And Procelera is one such bioelectric dressing. It produces electric currents similar to the physiological electric fields present in the body. It produces micro currents of the range of 2 to 10 microamperes. Electromagnetic therapy has also been used as a biophysical signal. It resulted in upregulation of multiple growth factors and including nitric oxide, which is very important for angiogenesis, and also this therapy has also been shown to regulate cellular migration. In the next few lectures we will look at different fabrication techniques that have been used to synthesize tissue engineered skin grafts.
So, as Vasudha discussed about the design and other characteristics for making skin graft that can be cumulatively made into three categories. The first one is freeze drying, and the second one is electrospinning, and the third one is injectable hydrogel. So in freeze drying, what they do is they make the polymer solution and they will keep it in the frozen condition. Based on the melting temperature of that solvent, it will get frozen and it will form the crystals. So based on whatever temperature we keep it, that crystal size will be formed, and based on that, that pores will be formed into that scaffold. So in electrospinning, they will be passing that polymer solution under the electric field. So when it is spinned, and that will be collected into that substrate. So the fibers whatever is collected, that will be forming a mesh like thing, and that can also be used as a skin graft. So in the injectable gel, in outside of the physiological environment, that will be in the solid form. When it is injected into that physiological environment, that will form the gel. So the advantages with the injectable gel over that freeze dried scaffolds and electrospinning based scaffolds are like that automatically fills that wound bed. We do not need any specific mold or something, and we do not need any other pre-processing. So what we need is, we just need the polymer or with or without cells, and that can be directly put into or injected into that place wherever that wound is, and that will fill that wound, and it will form the gel. So that wound bed will be completely covered. So another advanced technique which is involved with making skin graft is 3D printing. So in 3D printing, it involves various steps. So first what we need is, we will be scanning that particular tissue. So if we take skin, so that skin will be imaged under CT scan or MRI scan. So CT scan involves X-ray, and that image will be taken at all the 360-degree angle. In MRI, under the magnetic resonance field, under 360-degree the image will be taken. Once that image is taken, that will be converted into printable form like CAD format. So because then only we will be able to print that particular structure whatever we are interested in. Then the next step is material selection. So in the material selection, whatever criteria Vasudha discussed, we all need to go through all those things, and we need to decide what material we need and for what purpose we need. So all these things need to be decided. Then in case of cell selection, we have epidermis. That epidermis contains keratinocytes, and dermis contain fibroblast. So apart from that, we also can go for some stem cells, as she discussed. So like based on our interest, we need to choose keratinocytes or fibroblast, or together keratinocyte and fibroblast. It's up to us, like what we are going to make. If you want to make only epidermal graft, then we need to choose only keratinocytes. If you want to make dermal graft, we need to choose only fibroblast. Or like if you want to make that skin to be in that particular color, we also need to choose melanocytes, and that needs to be co-cultured with the keratinocytes. So it's all up to us that what we are going to make. Then the next step is bioprinting. So bioprinting can be of can be printed three ways. Like we can use inkjet printer, or like we can go for micro extrusion based printer, or like laser printing. So we have already discussed about that 3D printing techniques and how all these things works. So we don't need to go in detail about those things. So like we as we know that the hydrogels whatever we are going to make, that hydrogel should need to be biocompatible. Only that biocompatibility and gelling nature will not help to use for the biofabrication. So there are certain criteria that needs to be considered. So consider like that forms gel, but when you apply some stress or something, the cells will not be viable. Then it is not of no use. Like when we print, it is making gel, then also it will not be useful. So the printing fidelity and the speed when that gelation occurs, all these things play a very important role. So all these things are mainly controlled by two main parameters. One is viscosity, or the rheology of the polymer whatever we use for biofabrication, and the second thing is cross linking mechanism. So, in viscosity what happens is like viscosity mainly played by two parameters. First one is concentration of the polymer and the second one is molecular weight of the polymer. These are the two parameters decides the viscosity of the any polymer solution. So, if we take high viscous polymer with low molecular weight, what happens that nozzle will not be able to print that particular thing. So, if we use that micro extrusion based technique, so we use the needle, and that over that needle we will be applying that pressure into that needle. So, while we apply the pressure, that will be pushing that needle and that will be printing. If that solution is highly viscous, what happens, instead of making solution, it will form the droplet. So, to avoid that, what we can go for is we can mix it with some other polymer, or we can use less concentration of polymer with high molecular weight. So, we can go for this way or that way. So, in the left side we see one fine example. So, in the example what they have done was, in the first image A, so they used gelatin methacrylate, which is a known polymer. So, when they used only that gelatin methacrylate, it was not able to print, whereas it formed the droplet. But when they mixed that with the hyaluronic acid, what happens, it formed the clear solution, and once that solution is formed, they were able to get the structure of their interest. So, viscosity plays a very important role in controlling this. So, another thing is that shear stress. So, in the shear stress, if we apply more shear to the polymer solution, obviously the cells will get died. So, there are reports that even if we apply 1 Pascal of shear into that hydrogel containing cells, the endothelial cells get detached from the substrate. So, consider even 1 Pascal of pressure is applied, that cells are getting detached. It means when we apply that shear into that needle or whatever printing technique we use, the cells will not survive. So, we will get this hydrogel and we will get the resolution of our interest, but the cells will not survive. In that case, there is no point of doing 3D printing. So, in the same 1 Pascal of shear stress, they have also found that cartilage function is also lost. So, to avoid that, what we can do is we can increase the speed of the printing and reduce the shear stress. Say for example, we have the needle, and that needle we are applying some pressure into it. So, the time of applying that pressure will be less and the printing speed will be faster. So, the shear whatever is given into that particular polymer solution will become reduced. So, the number of cells what we get will be more, and that will be viable. So, that is the ultimate idea in this. And the next property with shear stress is that shear thinning. So, in the shear thinning what happens is when the polymer solution is in the needle or particular dispenser, so that will be in one structure, and when we apply some shear into it, that shear thinning will happen and the structure will be reformed or reorganized. That we will be able to see in the second part of that image. Then once it is printed, that forms particular hydrogel or the structure of our interest. So, again that reformed into that shape whatever we are interested in. So, if we apply more shear into that particular thing, that reorganization will be a difficult thing. So, we will not be able to achieve that reorganized polymer structure in the substrate. So, if we are not able to achieve that reorganized structure, the gelation and other things also will get varied. So, these are the main parameters we need to discuss or we need to know whenever we are going for 3D bioprinting based graft development. Then the next step is that gelation, that as we all know that gelation can be of physical and chemical. So, in physical we can use some ionic cross linkers, or that could be of electrostatic based interaction, or we may pass even UV or something and we get gelation hydrogels. And chemicals, like we can also use some cross linkers, because of that cross linkers that forms gel. So, these are the main parameters we should know before going for bioprinting. So, in case of 3D bioprinted construct, so as we discussed in the design criteria, we need to have the scaffolds. So, that graft can be of acellular and cellular. So, if we want to make acellular based scaffold or graft, what we need is we just need only polymer, and that structure whatever we need to print, that will be printed. If it is of cell based therapy, then along with the scaffold or hydrogel, we need to culture the keratinocytes and fibroblasts, or only keratinocytes or only fibroblasts according to our interest, and they will be grown in vitro, and once they mature, they will be placed into that patient body, and that will be used as a regenerated skin. So far with various techniques, they were able to achieve that particular thickness. So, laser based technique, they were able to achieve 600 micron around thickness of the skin. So, in case of microval based thing, they achieved about 150 micron. And like they have used various materials like collagen, hyaluronic acid, chitosan, and various type of biopolymers. Especially they are all water soluble polymers. So far very limited papers are available with hydrophobic polymers, and that to be used for skin graft applications. So, so far with the design and design criteria and other things, they have developed some commercial products. So, herein we are not just going to discuss about the skin graft based products. Even like if there is a wound, they will be doing some dressings. So, even that also will be we are going to discuss here. So, the first one is that hydrocolloidal dressing. So, that will promote that debridement. So, in the debridement, one of the finest product from that is Comfil. So, that was developed by Colopost Denmark company. So, what it does is that will promote debridement, and it will avoid that moisture to go outside. Then the next second thing is alginate dressing. So, that dressing is made of alginate material, and again this also will give this will act as a absorbent, and one fine product from that alginate dressing is Sand, that was developed by Maersk, Denmark based company. So, the next product is non-alginate dressings. So, in the non-alginate dressings, we have Nu gel developed by Johnson & Johnson. So, this will again provide that promote debridement, and it also gives that absorption to the wound site. So, now that Nu gel comes with alginate also, just to promote that hydrophilic nature and absorption. So, we have semi-permeable films. So, here we have bio-inclusive developed by Johnson & Johnson. So, herein we have polyurethane film. So, that polyurethane film is coated with acrylic adhesive. So, what it does is when we place into the wound, that will automatically adhere into that wound site, and it will pass that air to pass, and that will be keep maintaining that moisture condition. So, if it is dried, it will be difficult for that wound to heal. So, to maintain that, they use semi-permeable films. So, they have foam dressings also, as we all know that foams mainly made of that polyurethanes. So, this is just to protect from the thermal environment. So, from various temperature it can be protected. So, that is made of that polyurethane. So, we have antimicrobial dressings. So, as that name indicates, that dressing is to avoid that infection, whatever microorganism invading into that body, that can be avoided by these type of dressings. So, Acticoat is developed by Smith and Nephew Healthcare, and that is one of the product which is available. So, these are all the products whichever is available for simply wound dressing, not for the graft. So, when we come to the graft, as we told, like the graft can be of split thickness graft or full thickness graft. So, split thickness graft, even that could be of dermal graft or epidermal graft. So, full thickness graft, it will have epidermis as well as dermis. So, the grafts can be of acellular as well as cellular. So, all these things people have developed and they have the products. So, epidermal grafts, they are mostly of cell based grafts. So, few people use some scaffolds, whereas few doesn't. So, epicell and cell spray, they are simply cell based things. What they do is they only supply the cells into that wound and they will start growing. So, the my skin and laser skin, they have the scaffolds. So, here they use collagen and alginate based thing, and dermal substitute. So, this is here in that one of the successful product which is Integra, developed by that John Brook. So, we will be discussing about that, how that works. So, apart from that, alloderm, hyalomatrix made of hyaluronic acid, dermograft, transit and hyalograft, all these products are available. In case of full thickness graft, we have Orcell, Appligraft and Polyactive. So, the companies whichever is developed also it is given. So, as I told, Integra is one of the finest and successful products in the market. So, that develops only that dermis part. So, that involves dermis development by the scaffold and epidermis development by the autologous skin. So, at the first day, consider this is the wound. So, what they do is they clean that debris whatever is present at that wound site, then they will cut that portion wherever that wound extended. So, once everything is done, they will be placing that graft. So, that graft contains that collagen and hyaluronic acid as a bed, and over there we have the silicon layer. So, after 7th day that new dermis will be formed. So, once that new dermis is formed, they will remove that silicon layer whatever is present on the top. So, silicon layer is in general it is given just to avoid that contraction and to avoid that moisture to go outside of the wound bed. So, once that silicon is removed, they will take that autograft by dermatome or any other technique. They will just cut that tissue from the wound patient and they will place it over that dermis. So, after 25 or 56 day, we get the regenerated skin. This is how this Integra works. So, so far we discussed about that skin graft design and why it is required and what are the techniques involved in developing such skin graft. So far there are so much of study has been done and they have tackled so much of problem. Still there are some problem exists. So, the main problem is cost. So, if we consider that cost in US dollar, so 50,000 US dollar is needed just to cover that 40% of the wound for a male patient. So, this is highly costlier. So, to avoid that we need to go for some easy strategy to make some skin graft. Then obviously that fabrication techniques also a very limiting factor. So, even 3D printing is not an exception. So, in 3D printing resolution and other things play a role. So, this needs to be optimized so that we will be getting the proper skin graft. Then the next is number of cells. So, as she discussed like the choosing of cells and number of cells whatever we are going to encapsulate into that hydrogel, that matters, because if we seed very less number of cells, that will not proliferate and that will not do the function of our interest whatever we are interested in. So, if even we should not take higher the number, even if we take higher the number, then unnecessary fibrillation and other things will happen, because of that that wound will get spoiled. So, appropriate number of cells need to be chosen, and that also another limiting factor. So, as I told that in the anatomy section, like vascularization plays an important role, because of that vascularization only the cells are growing into the top direction, say in the epidermis, from epithelial cells to keratinocytes and further the next to next layer, and finally the cells are getting dyed and they form the shed of the cells. So, if that proper vascularization is not there, the signals or nutrients will not pass into one layer to other layer. So, that achieving vascularization also is happening by applying negative pressure and some electric field, as we discussed in the previous session, but still it plays a very limiting role in vascularization part. So, innervation is another important factor we need to consider, not just for skin. With all the grafts we have this problem. So, this also another limiting factor for this skin. So, once that successful skin graft is developed, we can use it for wound healing. So, that could be of that wound happened because of accident or burns or something, or that could happen by diabetic wound or something. So, also it can be used for screening various drugs that could be used for various cosmetic applications. Thank you.